looks like we've got Robin from North Carolina State University, Peter from APHIS, Alice from IR4, Clayton from USDA's Office of Pest Management Policy. So that's awesome. We've got Dylan, and Dylan is a market development specialist, agriculture at Bitech in Kansas, Oklahoma, and Texas. Got Roger as well, Lacey from Plant Diagnostic Services, and we from CIPM at NCSU. So welcome to everyone. We're certainly glad that you're here. And I think we are going to go ahead and get started. So again, welcome to the Southern IPM Hour on sorghum aphid. And it's presented by the Southern IPM Center, where we present research issues and programs in integrated pest management from the Southern region through this webinar series. This IPM Hour takes place typically on the first Wednesday of the month at 1 p.m. Eastern. And I am Kayla Watson. I am the Communication Director for the Southern IPM Center. We are housed at NC State and UGA with a mission to coordinate IPM across the region. We're also one of 4 regional IPM centers supported by USDA NIFA Crop Protection and Pest Management Regional Coordination Program. For the webinar today, we're actually going to be testing your sorghum knowledge as we go, just trying to give you a way to interact with us a little bit. So be on the lookout for those questions to pop up on your screen. And if you have questions for the speaker as we go throughout the talk, we'll be answering those questions at the end. So, or, you know, if it seems timely, we can also answer those as we go. So if you could just type those questions into the Q&A located on your Zoom control panel. The speaker that we have for you today is Dr. Raul Medina, and he's going to be speaking with us about the sugarcane and sorghum aphid invasion in American sorghum. And he's going to be talking about updates and future directions. And Dr. Medina is a professor in the Department of Entomology at Texas A&M University, where his lab conducts research aimed at understanding the modulation of species interactions among parasites and their hosts as a result of population genetics and evolutionary factors. He was here. Raul, are you still with us? Hopefully he will rejoin in just a moment. But Raul has had a couple of projects that have been funded by the Southern IPM Center. And I think that this presentation is a combination of those 2 projects. So we're really excited to hear about the research that he's been conducting, especially now since there has been confirmed a sorghum aphid. It was thought to be originally just sugarcane aphid, but as they were doing the research, they realized that it was a different insect altogether. So we're excited to have Raul here to tell us a little bit more about that. So welcome to the talk, Raul, and we're excited to hear about your research.
Thank you very much. So let me just share. Can you see my screen right now?
No.
Yeah, something happened. Let me just share it right now. Okay, there we go. Give me 1 second. All right. So can you see it now?
Yes.
Yeah, the computer kicked me out. Sorry, but I'm back.
It's okay. We're glad you're here, and that looks good.
All right. So yeah. So let's talk about this sugarcane sorghum aphid invasion in the U.S. So let me start by, first of all, reminding everybody that this is, I'm going to talk about a pest, and it's going to look like biology and systematics and all that stuff, genetics. But at the end of the day, we are trying to understand all these things to be able to perform IPM, which I don't need to remind the audience here what it is about. It's just a synergy of different strategies that we use together to combat pests in the most sustainable way we currently know. Just reminding you of this concept by using this infographic from the ESA, Entomological Society of America. So let me jump in about the issue here, so we have time perhaps for discussion at the end. So this is about the sugarcane aphid, or what we used to refer to as the sugarcane aphid, which is basically an outbreak in U.S. sorghum that occurred in 2013. We observe a weird aphid in sorghum, something that we have never seen before in that crop. Very quick, in 2013, after first reporting it, it was observed in several counties in Texas and even in Louisiana. So I will ask you guys to guess, how many states do you think are compromised today with this aphid in sorghum? There is a quiz there that you guys feel free to make a guess, and then we'll see how we do. All right. So show up in Texas. So in 2013, there were 2 states involved. So what do you think is the state today? I give you some time to put your answers there. So do I need to put that answer myself? I wanted to put it. So Kyla, is there, I mean, will you tell us the right answer, or will we be able to see it?
So the correct answer was 22. And you can kind of see how everybody was guessing. It's pretty even across the board, actually.
Very good.
22 states now.
So these people know what we're talking about here, because it's very close, right? As you can see, as of 2021, which is a couple of years ago, these have not changed in terms of the states today. This is the coverage of the aphid in sorghum growing regions in the US, right? So pretty quick. And this aphid basically took every relevant growing region in the United States. So this is a problem for us in sorghum. And at the beginning, let me see. Yeah, and then at the beginning, this was, like very problematic, because this aphid has the ability to occur in the thousands and overwhelm the sorghum plant pretty quickly. As you can see here in the pictures, this is how abundant they can be. And then the first time this was observed, sorghum growers got really concerned. Some sorghum growers lost almost 100% of their crop due to this aphid at the beginning of the invasion. The other problem with this aphid is that it produces a lot of honeydew, so much so that besides inhibiting photosynthesis, it also, the honeydew interferes with farm equipment, as you can see in this picture on the right. This is a mess. So looking at this damage, right, I asked you, the audience, again, we have another one of those pop quizzes. How much acreage of sorghum, or how much sorghum do we grow in the US? Anybody? Oh, already answered. 1 million acres of sorghum were planted in the US in 2021, right? So you can imagine that significant economic cost for the country by this aphid if all these problems are showing up. So at the beginning, we have never seen an aphid like that in sorghum. Oh, wait. Can you hear me all right, Kyla?
Yes.
Okay, perfect. Yeah, so we have never seen an aphid like that in sorghum in the US. So at the beginning, we were trying to name it, and it was concluded, and we have some pressure for do this naming, because imagine there are people, lifelines here, people growing sorghum were really concerned, and we needed to somehow have permission to use chemical control against this pest, or some kind of control, and chemical control popped to mind because it's the easiest and fastest to act. So it was important to have a name. The problem is that the identification of this aphid is not easy because it's part of a complex, and when we use the traditional ways to classify it, it was exactly in the middle of the classification. So we didn't know if it belongs to 2 potential species, sorghum, Melanaphis sorghum and Melanaphis saccharine. It was really right in the middle when you use morphology of individuals. So it was decided to use the name Melanaphis saccharine, the sugarcane aphid. And we can talk at the end why was that decision made, but it was made. And when that happened, well, that's good because now we have a name. But the issue with the name is that it was strange for some people at the beginning, because the Melanaphis saccharine the sugarcane aphid, has been reported in the US in sugarcane since the 70s, since 1977 in Florida, and since the 90s, late 90s, almost 2000 in sugarcane in Louisiana. So we had this aphid in the country before. And hint, that's in part why this name was chosen, because if in doubt, we prefer this name for reasons we can discuss at the end. But this was a name given, which was great for applied purposes. But then, of course, people interested in the biology of the aphid got a deeper dig into this question, right? So the first issue to jump to mind is like, you wonder why an aphid that had been in the US since the 70s in sugarcane all of a sudden decided to jumped to sorghum in 2013, right? So that's one question that was in the back of a lot of people's minds. The other thing is that when you look at the host range distribution, the host range of the sugarcane aphid, it has a wide host range, and it has reported to attack several plants, including corn and rice. So at the beginning, of course, when this name was given, some people was really concerned, and one of those was me, and we were like, what if this thing jumped into corn, into rice? And several people, not only me, but mostly people like David Kearns, Scott Armstrong, started trying to test the host range of the aphid we got in the US in 2013 and see if it could jump to corn and rice. And then good news, it didn't. It performed really poorly. Actually, couldn't even feed in corn, but it did fed on Johnson grass and other varieties of, and several varieties of sorghum and miscanthus and other wheats, but not in the moneymakers, corn, rice, and things like that. So that was good. That it seems that the local host range of this aphid in the US was reduced compared to the reported host range all over the world for this aphid. Okay, so people like David Kearns and Gregory Wilson, his PhD student at the time, started to do these tests of what this aphid can feed on in the US, like the aphid that just was show up in sorghum since 2013. And they exposed the aphid to several plants. Here you can see I have highlighted sorghum bicolor and Saccharum officinarum, which is sugarcane, and then some plants in the middle, like Johnson grass and miscanthus, right? The thing I want you to take from this slide is that for every single fitness measure in this table, the aphid in sorghum performed way better on sorghum than in sugarcane. And this was interesting because the extreme of better, right? This was a little bit, it makes some people suspect that if this biotype just show up, it was extremely good at attacking sorghum and terrible at attacking sugarcane, which is something that you will not expect in a recent host switch. But that was evidence we have. So we asked, well, let's test, was this really a host plant switch? You know, the aphid we got in sorghum is the same species that we used to have in sugarcane, yes or no? And to do that, we sample this aphid from sorghum and sugarcane from all the places that they occur at the time. This is 2014, 2015, 2016. And then we got samples from South Africa just to compare, because we knew that in South Africa, this aphid had been on sorghum for at least 30 years. So we sampled from those places, and this is what we found. Well, no, this is not what we found. This is what we knew before the invasion. And bear with me, and then I will just make sure, ask Kyla. Kyla, can you hear me all right?
Yes, you sound great.
Okay, good. Because this is very important. I want to make sure everybody hears it. This is what we knew about the genetic composition of this aphid before the invasion in 2013. Every one of these circles represent a different genetic haplotype of this aphid Melanaphis saccharide in the world. And as you can see, they are color-coded. And the one that matters to us is here, the sugarcane aphids, Melanaphis saccharide, that was present in the US in the 70s and in the 90s, was mostly found in Louisiana, Florida, and Hawaii, belongs to this purple, we call it multilocus lineage, that's what this stands for, MLL, multilocus lineage D, right? Very different to the multilocus lineages of the sugarcane aphid that we found in other regions of the world, all of them attacking sugarcane very heavily, right? So that's what we knew about the sugarcane aphid before the invasion in 2013. So after the invasion in 2013, this is what we found. This respects the same color codes that I just showed you, it's just that it doesn't show the map. But the one that is important to us is here, the multilocus lineage D, which is the sugarcane aphids or Melanaphis saccharide haplotype reported for the US before 2013, is observed here in these circles that represent the multilocus lineages as told by microsatellites The squares represent the mitochondrial haplotypes as told by CO1 gene. So as you can see, this already, for people that know genetics, they already know the answer to the host switch question. Because as you can see, the M here in yellow, you see the multilocus lineage to which the aphid that invaded sorghum after 2013 belonged. Very different than the one we used to have before. So this MLLF is the multilocus lineage that groups all the individuals we found in sorghum in the US post 2013. And it belongs to a different mitochondrial haplotype, H1 here. So the distance between these two haplotypes, both mitochondrial and nuclear, is so vast that it's very unlikely we are talking about a host shift. In a host shift, you will expect the new one to be very close to the original one. So it's obvious from this genetic map that we got that from somewhere else, right? So, and I will show you the graph again, but I want you to keep this in mind. When you look at the multilocus lineage F, which I have circled now in this black circle there, if you measure the genetic variation of this, you will realize that within that multilocus lineage, you do have further genetic subdivisions or variation. We found 3 genetically distinct genotypes of this multilocus lineage in the US, but they didn't associate or corresponded to location or geography, which is represented here by the initials of each state in which you sample it, or by host plant, which is represented here by the different colors from different host plants. So what does this mean? It means that the multilocus lineage F in the United States have genetic variation within, but it doesn't associate or group based on host plant or location, which is interesting because it's rare for a host shift to have so much genetic variation at the beginning of its origin, right? So most likely this, again, points to the hypothesis that we got this from somewhere else, perhaps in more than one introduction. That's what this is telling you. So let's say, so host plant, the answer, is this a host plant switch? The answer is nope. A host plant switch does not explain the occurrence of this new aphid in the US sorghum for the genetic, because of the genetic evidence I just showed you. There was no geographic population genetic structure detected, which is strange for a thing that just showed up, and no host-associated genetic differentiation detected. So no biotypes per host plant. But there was interspecific genetic differentiation detected, right? So that's some piece of evidence that I want you to keep in mind. Then we were asking, okay, but the thing switched. So we know that, okay, it's not a host switch in the genetic sense, meaning that the insect we observe in sorghum didn't genetically evolve from the one we used to have in sugarcane in the US, but it definitely appeared in a new plant. Could it be due to a bacterial symbiont? Could a bacterial symbiont be involved? And I ask this question because I want you to keep in mind this sentence, that when invasive species show up or arrive to a new crop, they do not arrive alone. They usually arrive harboring a bunch of microorganisms within themselves and on their surface, right? And I'm going to ask another one of those pop quiz questions to you guys. Answer this question, and then we'll move on. So the absence of this organism has been shown to cause sorghum aphids to die. Which of these ones do you think that is? If they don't have this organism associated with them, they do not do well. Can somebody guess? And Kyla, you will need to tell me the answers because I don't want to answer it, because then I will bias the answers. Or maybe we'll answer after a little bit. How about now? I can't. Okay.
Let's give them just a couple more seconds.
Okay. So you will need to tell us the answer. Oh, sorry. And I will drink water while I wait.
Let's see. Got about half of the participants that have taken it so far. Okay, we'll go ahead and end it.
They are Googling.
They are. We can't give them too long to Google. Okay, there's the results, and you can see that the correct answer is the bacterial mutualists. So
bacterial mutualists are extremely important for several insects, but it's in particular to aphids. Aphids happen to have primary and secondary bacterial symbionts, and without the primary ones, they do not survive. In the case of aphids, they do share almost every aphid in the planet have a Bugnera fidicola or aphidicola, depending on how you pronounce it, as its primary symbiont. Without that symbiont, the aphid dies of starvation because he cannot get the amino acids it needs to survive. So bacteria are very important for aphids, and then they have secondary symbionts that can do a bunch of interesting things for them. Like in this case, for example, this is not an aphid, but it's an insect, the kutsubac. And in this aphid, the secondary symbionts associated with this guy make it do interesting things. In here, you see the bug and then the X. And I don't know if you can see in this tiny picture, but there are some arrows there. Let me make it bigger. When you see some of them point to the phrase that the insect adds to the X, and in that phrase, the phrase has a bacteria, Candidatus shikawella, which makes the kutsubac change host plants. So basically, Candidatus shikawella capsulata produce this thing that I'm about to explain. So when you have the kutsubac attacking soybean, you measure and you test if they have Candidatus shikawella capsulata. They do. And if you switch the bacteria and place it in a bug that is only feeding on kutsu, that cannot feed on soybean, if you do that change, all of a sudden, now that individual, the same individual that before couldn't feed on soybean, becomes a soybean feeder. So we do know that some bacterial symbionts, particularly secondary symbionts, can change host switch. So we were wondering, is it possible that something like that could have happened in the sugarcane aphid, making it be able to eat sorghum just because he got an interesting bacteria? So we had that question. We asked that question, right? Could bacterial symbionts be responsible for the aphid-pest outbreak in sorghum? And to answer that question, we sampled the aphid again from the same places we sampled before. Basically, we had all those samples, so we are like, why don't we measure these with the samples we have? So they are the same places we have aphids before, and we got tons of aphids, so we could do more than one study with those. And I think at the end, you will see a huge list of names, and those are the people that send us aphids from all these places. We didn't go to all these places ourselves. Okay, so we collect from there. And then we did two things. We did a traditional diagnostic PCR, which is basically testing using primers of known bacteria that are known to occur in aphids to see if they are there in the sorghum aphid. But then, because this is an aphid that we were not very familiar with when it comes to bacterial composition, we also use high throughput sequencing to see if there were unknown things there that we need to look at. So I will show you the results right away. So when we look at the PCR results with the primers of the usual suspects, nothing surprising. This is just a pool of the aphids we got from different states and different host plants, and in all cases, we found Buchnera aphidicola, as this is showing. This is not surprising. That's what we were expecting. All aphids have that. Now, the surprising part comes here when I'm only going to show you one of several slides that look exactly the same. So instead of boring you, showing you one after the other, I'm just showing you one, and I am telling you that all the tests we did for 6 common usual bacteria that are found in other aphids yield this result. In this graph, you see the gel. The first letter here is in each of these columns represent the state, and the second represent the crop, so grain sorghum, and then Johnson grass, and then sugarcane. And as you can see here, we have a positive control here, meaning that it's a sample that we knew was full of Hamiltonella, which is a secondary symbiont in aphids, and none of the samples, and these are pooled samples. So each one of these columns grouped together like at least 30 individual aphids from different locations, and different locations within those states that is showing, and not a single Hamiltonella was found. So that's weird because none of the usual suspects were found in this aphid. And let me summarize that information by using this table. Here is the sugarcane aphid, our creature that we're discussing today. And on the other columns, you see other aphids and the bacteria that you traditionally find on them. Like Buchnera aphidicola, I told you, is found in all of them. Hamiltonella defensa, which is the slide I show you, is not shown in the sugarcane aphid, but that's not too weird because the green peach aphid doesn't have it either, but several other aphids have it. The weird part of the sugarcane aphid is that none of the usual suspects are found in this aphid. That's interesting, but it's not that surprising because very few people have used very few models to study the microbiome of this aphid. So it's just a call for more studies on aphids that are not the traditional ones, because as you can see, not everything you found in the P aphid is going to be found in other aphids. Particularly now we move to arboreal aphids that are completely different than this one that is herbaceous, right? So that's my just pay attention if there are graduate students in the room. There is value in exploring things that have been explored in model systems in new species. But okay, so that's what the PCR results with the primers of the usual suspects tell you. But how about the high throughput sequencing part? That's when you test things that are unknown, right? Okay, there we found a bunch of interesting things. Almost all bacteria here in this graph that I'm pointing in the pointer here, I don't know if you can see the pointer moving up and down, but in the basically in the y-axis here, everything but Bugnera is new in aphids, or almost new in aphids, except for Pseudomonas, perhaps not. But as you can see, there are two things I want you to see from this graph, or to take from this graph. So first of all, that there are very unusual several unusual bacteria that you can detect with high throughput sequencing that you will have missed if you just use the primers of known bacteria. That's the first take home. And the second take home is that there is some variation in bacterial abundance based on host plant, as you can see here in Arcobacter, Bugnera, and Citrobacter. The important ones in this being Arcobacter and Citrobacter. If you are this aphid and you like to feed on grain sorghum, you will have way more sequences of Arcobacter than sugarcane, than the ones that feed on sugarcane. And if you happen to be an aphid that like to feed on sugarcane, you will have way more bacterial members of the Citrobacter group than if you feed in grain sorghum. Now, this is relevant because Citrobacter in other insects, like in this fly here, have been shown to detoxify insecticides. So there are some valuable practical applications, potentially practical applications in understanding unknown components of the microbiome of aphids, right? Another thing for you to think about. Now, the other interesting thing here is that when you look at the bacterial community as a group, not by individual player, you see that there are significant differences based on host plants. So basically what this graph is telling you is, here you see the shapes represent the host plant, which in this case is grain, sorghum, and sugarcane, and the color ones here represent states. C is supposed to be California. Sorry, I'm missing an A. But here what this graph is telling you is that there are significant differences in bacterial community composition between things that feed on grain, sorghum, and sugarcane. So you have the same individual players, but the different in abundances among different host plants kind of produce a different kind of bacterial community. And that's important to keep in mind. Let me just check. Okay. All right. So that study comparing the different microbiomes of aphids in different host plants, we published in Annals of the Entomological Society of America. And I like to show this just because it's the first time in my life that one of my papers make it into the cover with one of the pictures of our group. So I'm very proud of this picture. So I just wanted to show it to you. So that's the only purpose of this slide. But all right, so if we ask the question, can a bacterial symbiont explain the 2013 outbreak? The answer is no. But bacterial community is different in sugarcane and sorghum, which is an important detail to keep in mind. So here we have that we don't have a host switch. We don't have a bacterial explaining this switch to sugarcane. So what is it, right? So now with the information I have given you so far, that it can be summarized in this slide by, variation in the traditional host plants that you will expect from the sugarcane aphid, melanophis variation in bacterial community composition between these two things. When this aphid will feed, it's supposed to be the same species, but you have different bacterial compositions on different host plants, sugarcane versus grain sorghum. And then the fact that the genetic distances are so far from each other, we start wondering, could it be that in fact we do not have two populations or two multilocus lineages of the sugarcane aphid melanophis saccharide, but in fact we may have a situation where we have a cryptic species. In other words, are aphids on sorghum and sugarcane cryptic species in the US? And that's the question I'm about to explore with you guys in this series of slides I'm about to present. So for this one, we collect samples from all over the world from two of the host plants, sugarcane aphid and sorghum aphid, right? And I remind you that this is what we knew before, when we did the study before, this is what I show you repeated times. I just want to remind everybody that the multilocus lineage that define the traditional sugarcane aphid in the US is here, and the one that is the new invasive aphid in sorghum is here, right? So that's what we knew. So in this new study, we tried to do a couple of extra things just to be sure. In here, you can see the same multilocus lineages grouped together, but what you see in blue and in yellow, both in the circles here and in the structure graph here with the lines, is the diet, right? So in blue, you see things that do very well in sugarcane. In orange, you see things that do very well in sorghum. And they map perfectly. When you look at the genetic composition, multilocus lineage A, multilocus lineage E and F group all together and do very well in sugarcane, while the other multilocus lineages group together in the structure graph here in orange, all here, B, C, and D, right? And they do very well in sorghum. So interesting that these guys are far from each other. You know, like the one in sorghum and the one in sugarcane in the US are far from each other, and the distance, the genetic distance corresponds to the variation in diet. The other interesting thing I wanted to show you, when we use together CO1, CO2, which are mitochondrial genes, and elongation factor 1 alpha and interspace, ITS2, right, nuclear genes. When we do together a multivariate analysis considering the evidence that all these genes give you together in a multivariate fashion, we can separate the same thing that I just showed you with the microsat. So they come with the same picture. In a multivariate fashion, you can separate cluster 1 and cluster 2, the groups, the same multilocus lineages as before. And interestingly, although we failed to find diagnostic markers in mitochondrial genes, we did find a diagnostic marker in the elongation factor 1 alpha gene. If you have a T in this position, you definitely have Melanaphis saccharide, the species Melanaphis saccharide. If you have an A in this position, you have Melanaphis sorghum. And we tested this with the ones that we were sure were those, and then with the ones in sorghum that we found post-2013, and invariably, almost every single individual we collect from sorghum post-2013 in the US, even in Louisiana, have an A at this position, belonging to Melanaphis sorghum. So that's what the molecular stuff is telling us. Now, how about the morphological stuff, the morphometrics? For morphometric determination of this aphid complex, we use the ratios of these anatomical features that are pointed here, the process terminalis, the sifonculi, the cauda, and the hind tibia. And it's the comparison of these ratios that tell us, traditionally, if we have Melanaphis saccharide or Melanaphis sorghum. As I told you at the beginning of this talk, when we did this at the beginning, when we were rushing, desperate to help sorghum growers, when we did this with individual aphids, they were kind of always in the middle, in between what is supposed to be called Melanaphis saccharide and Melanaphis sorghum. So since we didn't know what to decide, Susan Halpern and Dr. Miller decided, Dr. Miller and Dr. Halpern, which actually worked together with me in these projects from the beginning, and I'm very thankful to them because they provide valuable specimens and information. They decided to go with Melanaphis saccharide, which I think was a good decision at the time. However, when we look at these things together in a morphometric, in a morphological fashion, and we look not at one, but several individuals collected from different places, different locations, different host plants, when you look not at individual ratios, but at a conglomerate of ratios in a multivariate fashion, we find separation of statistically significant separation between cluster 1 and 2, which again corresponds to things that we find in sorghum and sugarcane. They get separated when you look at that multivariately. And if you want to do by component, this is how the ratios look like when you aggregate the populations together. So although you cannot see discrete separation per individual, you can see continuous separation when you use populations of individuals. So here, for this ratio, and for all the ratios, you can see a statistically significant difference between the aphid that fits on sugarcane and the one in sorghum. And they just happen to correspond molecularly to the ones that map to sugarcane aphid, Melanaphis saccharide, and to sorghum aphid that maps to Melanaphis sorghum. And it just happened that everything we collect from sorghum in the US post-2013, except for Louisiana, that still have both, but everything else is maps to these blue lines here, Melanaphis sorghum. So we concluded, based on this evidence, that we are dealing in the US not with a different multilocus lineage, different population, but actually with an invasive sorghum aphid, basically Melanaphis sorghum, and we are trying to determine from where. We suspect that it could be northern India, southern Pakistan, but we couldn't get sample from there, so we cannot tell you for sure. And Dr. Samuel Nigush is actually very involved in trying to determine from where exactly we got the invasive aphid, but there is nothing conclusive at the moment. So let me see how much time I have. Oh my God, am I running out of time? Oh my God, Kyla, how much time I have? Because I want to tell you about another very cool thing, but I don't know if I have time. How much time
You've got plenty of time. You've got at least 10 more minutes if you'd like to keep talking. So you're fine.
Good. I love to keep talking, but I will also like to hear from the audience. So I will try to be quick on this one, okay? So the important thing I want to keep is a cryptic. This is important because cryptic species may differ in traits that are relevant to their control. And remember that at the beginning of the talk, I told you that this is all these investigations are done to help control this pest. So all these strategies that we have listed here as part of IPM actually work best in the populations that affect our crops in particular locations, right? So it is not news for everybody here in the audience that insecticide resistance is a great challenge for aphid control. As this graph shows, here you have time, and here you have how many instances of resistance we have to different chemical products. As you can see, they accumulate very fast. We are now in a kind of like happy place with neonicotinoids, but this red thing is maybe gonna increase over the years. So there is some pressure to use natural enemies to reduce aphid abundance. And we know that in some systems like the one represented here in this graph and in these pictures, in some places and in some crops, natural enemies work really well. And we also know, but maybe this is not very widespread, but some people is very aware that honeydew attract parasitoids in several systems. These have been proven, like in here, you see this parasitoid in this picture is attracted to this psyllid, as it can be seen in this picture that shows the trajectory of the parasitoid when you have honeydew in one side of the petri dish versus not honeydew. So as you can see, they try to hang out where the honeydew is. So we know that. So host plant can modulate honeydew production, as seen here in this graph for the pea aphids, for example, feeding on these two diets. You can see that the concentration of honeydew, the concentration of sugars in honeydew is higher when feeding on red clover than on broad bean, right? I just want you to keep that in mind. And also to keep in mind that these different sugar concentrations affect, and perhaps even compositions, affect the attraction of these honeydews to this natural enemy, this hoverfly, right? So here is just to show you that honeydew may have an impact in biological control. So what's the story with the sorghum aphid? So we know that this aphid occurs in sorghum and in Johnson grass in the US. And there is, we have casual observations that for some reason the natural enemies in Johnson grass seem to be less abundant than in green sorghum. And the same thing with parasitoids. We can talk about this more at the end because it's more complicated than this slide will seems to suggest. But we wanted to know, is the honeydew maybe involved in this? Because is it possible that Johnson grass and green sorghum modulate the honeydew composition or the abundance in a way that affects attraction of natural enemies? So we asked these three questions. Does host plant diet modulate honeydew concentration? Does host plant diet modulate parasitoid preference? And does honeydew age modulate parasitoid preference? Meaning that if you just accumulate a honeydew for 24 hours, will that change in 5 days or in 7 days? Will the composition of attractiveness of the honeydew change over time? So to answer that question, well, basically we place aphids in clip cages in these two host plants, and I'm gonna go a little quick on this so you can have time for questions at the end. And then we did two things: measure the concentration of honeydew and then perform a bioassay. And this was, when I say we, everything I do is done by my students. So this is my former PhD student, Crystal Wright, which is now a consultant at McKinsey in Boston. But so she switched from science to business, but that's fine. So here we have the results, right? So we found that in Johnson grass, we have more honeydew is more abundant in concentration of sugars than in grain sorghum. So interesting, because similar to what we saw with the host plants of the pea aphid example I just showed you. And then we wanted to know the preferences. So we use parasitoids that were reared on Johnson grass and in sorghum to make sure that they didn't have biases based on the rear environment. So we're going to call the ones we rear in Johnson grass the Johnson grass parasitoids. They are both Aphelinus nigritus, but this was reared in Johnson grass. Sorry, I don't know why it says S. It's supposed to be G. Johnson grass parasitoids, and then these parasitoids are reared on aphids that were feeding on sorghum all the time. So we call those sorghum parasitoids. Then we expose them to aphids. Aphid honeydews produced by these two different aphids on two different host plants. So what we did is perform choice tests, first with water, just to see if there was attraction or repellent to this honeydew. And then we measure preference of the parasitoid as a proportion of time spent on one treatment over the total time spent on both treatments. And then we conduct these bioassays at 24 and 120 hours. And this is going to be important for the graphs of results I'm going to show, okay? So we perform three choice tests. The first one and the second, it was just against water, so grain sorghum versus water, Johnson grass versus water. And the third one was Johnson grass versus honeydew to see which honeydew is preferred by these parasitoids. And this is what we found. So in here, in the first set of graphs here, you see what happened with grain sorghum parasitoids after 24 hours of honeydew production, and then what happened with Johnson grass parasitoids after 24 hours of honeydew production, and then the same thing, but now 120 hours. And as you can see, in every single case, the parasitoids prefer the grain sorghum honeydew over water. Basically, the honeydew produced by aphids fed on grain sorghum was preferred over water, which is good, which means the grain sorghum honeydew is more attractive than water. With Johnson grass honeydew, very similar situation. They prefer it over water, except for after 120 hours, there was no preference. So in this case, for some reason we still don't know for sure, honeydew of aphids that fed Johnson grass lost its attractiveness when it became old after 120 hours. Important thing to remember. Now, when we compare both host plants, here you have grain sorghum in purple and honeydew in green, you see that there is variation, and this is very useful and complicated, so perhaps you're not going to understand why, but that's okay. The important thing to take home from this slide is that it's complicated, right? But let me walk through this slide with you. So grain sorghum parasitoids after 24 hours prefer Johnson grass honeydew. Johnson grass parasitoids after 24 hours prefer grain sorghum honeydew. There are several hypotheses based on biology that we can make about this, and we can talk about this at the end. But when you have grain sorghum, all grain sorghum honeydew and all Johnson grass honeydew, in both instances, the honeydew from Johnson grass fed aphids was preferred. Okay, so that's what we found. There are some preferences. That's the good news. There is preference, and it varies over time, and it varies by host plant. And it gives us some tools to work when it comes to incorporating these things into biological control. The last thing I'm going to talk about, and I don't know if I have time, Kyla, do I have time, or do you want me to go to questions? I am happy to go for it. Okay, good. So one of the things we ask ourselves is that, well, there are microbes everywhere. I just show you there are microbes inside the aphid, but there are also microbes on the honeydew. And some of these microbes might be produced by the aphid or planted by the aphid itself, or some of them may be taking account of the environmental bacteria and providing a growing environment for them to thrive. Either way, this might have some ecological implications. So we wanted to assess the microbial composition of sorghum aphid honeydew. And to do that, we wanted to do that because we knew that some bacterial players, like Staphylococcus in honeydew of some honeydew producers, attract hoverfly predators, right? So natural enemies. So we already know that in some systems, a specific bacteria can be very important in attracting natural enemies. So we wanted to see what's the story with the sugarcane aphid. So we with the sorghum aphid. You see how confusing. I still call it sugarcane aphid. It's supposed to be sorghum aphid. Okay, so we collect aphid honeydew from Johnson grass and sorghum, put them in these not very fancy clip cages, and collect the honeydew in these pre-weight discs to see how much we got. And when we did a bunch of things that I don't have time to present here, we measure the amino acid composition, sugar composition, and organic acid composition, and compare them. That's something we can talk in another talk. But at the end, the important story is this. When you look at the bacterial composition of the honeydew in grain sorghum and Johnson grass, we didn't find any significant differences at the community level, kind of the opposite of what I show you for the bacterial composition inside the aphid, right? On the honeydew, we didn't detect any differences at the community level. However, when you see at individual players, these different columns are different times. Right here would show 24, 72, 120 hours, and 7 days. And then when you look at host plant, which I'm not showing, but trust me on this, there were 3 bacteria which bacterial composition changed depending on host plant and age of the honeydew. Actually, well, I don't count Bugnera, but let's these three. Bugnera I don't count because basically what is happening here is reflecting the abundance of the other ones. So there is a reduction, so they're correlated. So I'm not going to talk about that. But these three are interesting. And one of those is Staphylococcus, which is a bacteria that has been proven to attract natural enemies, as I showed you before. So there is something going on here. And we suspect, we still don't know what's the story with fungi in honeydew. We know that sooty mold is a problem, but perhaps there are fungi there that might be performing some ecological services for this aphid, for the sorghum. We don't know yet for who. But why is this important? Because now we know the bacterial communities on the honeydew and the bacterial communities on the aphid. And the bacterial composition was not explained, did not explain parasitoid preference though. So that's it, the identity of the bacteria we found didn't explain those variations in preferences in the parasitoid I show you, but perhaps microbial metabolites or microbial volatile organic compounds might. And that's something that we have not measured, but we are gonna measure, hopefully with the help of Angel Helms in my department. We're gonna write a project when we actually want to identify which volatiles and metabolites are produced by these bacteria in the honeydew and see if they have a role to play in attraction of natural enemies. So, and then the story I didn't have time to tell you for sure because Kyle will kill me if I tell you, but it's the fact that honeydew also attract ants. And we do know that this aphid, even though it just show up in the US in 2013, is already establishing interactions with Tony crazy ant, which happen to occur in several of the areas in which we have the sugarcane aphid, as shown in this graph. So there is potential there for a synergistic interaction between these players that doesn't seem to be promising for sorghum growers in the US. So hopefully I'm wrong, but we for sure need to explore this. And I have some data on this, but I don't have time to share it with you guys today. So the take home of the entire talk, I guess, is that complex challenges require integrative solutions. And for integrative solutions, we need to have a bunch of evidence from all sources. Microbial composition of, I have shown you just some example when we need the microbial composition of the honey, the microbial composition of the bug, when we need systematists, when we need ecologists, when we need geneticists to just make sure that the ways we have to control these aphids are more effective. And with that, I would like to thank people that funded this research. All the ones that send me collections are named here, and I may be forgetting a couple that send me just 1 or 2 vials, but I mentioned them first because without them, we would not have been able to conduct these studies. And I also would like to help the Southern IPM Center for funding for this project because they were the seed for all the things I have shown you. And then there were other funding agencies that jumped in for different components, funding students and the international component of this research I just showed. And everybody else that was involved here that is listed in there. I hopefully have not forgotten anybody. So with that, I would love to have some questions if you have any, or discussions or thoughts, anything you want, please go ahead and tell me, or Kyle, I guess, will read the questions in the Q&A, whatever you want. Thank you very much.
I really love that you show that at the end, Dr. Medina, because one of my questions was going to be, how many people does it take to make this research happen? So that's amazing. And I'm sure that, not all of those people are in Texas, right? So it has to go across state lines.
Oh my God, across continents, because my collaborator, Samuel Nibush, is in Réunion Island. He's part of the French government. That's why CIRAT is there. And the Department de la Réunion and all those, they were instrumental in helping us secure samples pre-2013 from Louisiana because they have them in their labs. So that was very useful.
Oh, wow. That's amazing to me. Just how much the team is, I don't know, how hard it is to put a team together and how many people are working on these projects. I did want to mention that you did kind of get a shout out from Lacey. Lacey says that you may have just cracked a question that they got from an entomologist working on pecan weevils. So with your research, so that's pretty cool. And maybe Lacey can let us know what she means by that in just a minute. But so definitely a compliment to your work there. And then we have a question from Alyssa, and this one is a little bit long, so it'll take me a minute to get through. And they say that you're doing fascinating work, which I definitely agree. They say, does the clarification on species inform the migratory history? So has sorghum aphid been in the United States a long time previously, or is this evidence of a new arrival? And if it has been here, is there also evidence that there's a new biotype since outbreaks didn't occur prior to 2013?
Yes, thank you for the question. Actually, it's a new arrival. We have not had records of the sorghum aphid in the US, as far as I understand. Not significant, definitely not being a pest, and I don't think we have a single record. So it's a new arrival. And I think maybe Clayton can help us out if he feels like talking. Clayton Myers can help us with this because he's familiar with exception and 18 from the EPA. If he's still in the floor, he maybe can tell us, because I believe that was instrumental for the naming, because we did have sugarcane aphid in the US since the 70s. So to get the exemption, it matters if you have already the insect in the country and we're just changing the host plant, so you can get that exemption quicker. But if we say that this is just, if we will have say this is sorghum aphid at the time, perhaps we'll not have gotten that extension that quickly. But I'm not 100% sure about this. Clayton, if you're in the room and you feel like talking, can you tell us? And if not, we can all kind of like ask that question ourselves to somebody that knows. But I think that's why Susan and Halpern and Dr. Gary Miller basically were like, oh, let's name it this way, because if we don't know either way, this will be more useful for growers. And I think that was a great decision. Give us time to do the research. That's what I think. But anyway, maybe Clayton is not in the room or he's busy. But yeah, but that's my hypothesis, let's say. But yeah, it's a new arrival, Elisa.
And Clayton, if you do decide that you want to talk a little bit more on that, just let me know and I can actually give you permission to talk if you'd like. So just let me know either in the chat or somewhere else, and we'll get you talking a little bit more about that. Let's see. I'm going to go ahead and allow you to talk if you'd like. There you go.
Oh, that's nice.
Yeah, and I apologize. I used Alyssa's link to log into this meeting, so I'm showing up in chat as Alyssa, but this is Clayton. And it was me who asked the question. So I appreciate the clarification that this was a new arrival. And my recollection, though, at the time in working on the Section 18s was that at that time, the hypothesis was that it was a new biotype. So even though it wasn't an invasive species, it still was given, very high priority for the Section 18. And it's really interesting to learn now it's not a biotype, it's actually an invasive species. I'm not sure if that will impact reauthorization of the Section 18s going forward, because now it's been extended to so many states and I think multiple active ingredients as well. So happy to connect with you after if you have additional questions on Section 18, but this is really fascinating. And I guess since I'm unmuted, I'd be curious if you think the Section 18 authorizations have been effective, or do you feel like this is the tools that growers have been given have allowed them to get ahead of this pest, or is it still really a big outbreak situation year to year?
Oh no, it was a lifesaver, because I think that once they start using the approved insecticides at the beginning, I think it was Sivanto or I don't, and the other one, I forgot the name, the commercial name, but no, it basically made 2014 and 2015 better than 2013. Now, I think we're getting a grasp. We're really heavily studying the potential for using natural enemies at the beginning of the season, and also new varieties having developed of sorghum that are kind of sorghum aphid resistant, and a combination of those, it's improving the situation. But at the beginning, we didn't have anything but those chemicals, so it was a lifesaver. So I think, and it was based on science, because we didn't know what I have just told you. It's very easy to go, back and say, oh, it was the wrong name. It wasn't the wrong name. It was the names of insects are all hypotheses of every organism. So the hypothesis with the information we have at the time was correct. And I think that the exemption really saved growers' lives. So I think it was very useful. Now, because this chemical happens to affect also the sorghum aphid. So
well, it's great to hear. And it's also, I certainly know that aphid taxonomy is very challenging. That's not my bailiwick whatsoever. I can't imagine. It's such an art form to clear the specimens and get them mounted properly and make all those tiny little measurements. My hat's off to everyone who helped crack this mystery. It's really impressive.
Well, that was Gary Miller, Susan Halpern, and then Samuel Nibush that did all that work with my student Justin Holt, helping some of them. So yeah, Samuel mostly. But yeah, I don't know how to do that either, Clayton.
So, Dr. Medina, you did say that you're okay with staying over just a few minutes if anybody else has questions, right?
Oh yeah, sure, sure.
Okay, so I did just want to mention, as people might have to leave, that we definitely want to thank our speaker today, Dr. Medina. And I put his email address for you in the chat, and I'll also put that in a follow-up email to you guys if you have any more questions and you have to go. Feel free to stay though if you'd like. And as you leave, there's going to be a quick pop-up survey after the webinar ends, and it's 4 questions, and that's going to help us guide the future of our webinars if you wouldn't mind to fill that out. And it should just pop up right in your internet browser. But again, you're definitely welcome to stay and keep talking. And Lacey, I don't know if you have some time to stay, but you were talking about, you know, Dr. Medina's research being pretty instrumental in a question from your entomologist about the pecan weevil. So if you want to talk about that, I can open that up for you as well. I've given you those permissions to talk.
Oh, that's fine. Thank you. I admit this is a now that it's anybody who doesn't find this interesting can leave. But as the sidebar, my father was a USDA ARS entomologist for 35 years, and yeah, you're all going to be scared now, but he retired and he started working on he went to work on the family farm, and lo and behold, you can take the entomologist out of the lab, but you can't take the science back out of the entomologist. So he has 20 years of data on pecan weevils and has proven all kinds of things about their life cycles that they didn't have before. But so this is an old orchard, and in wet years, it floods every year. And he has been putting traps on the same trees now, year in, year out. And yeah, we get to talking about his data because apparently that's a thing you do at Christmas time when you're an entomologist. And he's asking me about why is it you think that when the first flush of larvae are coming out, they're crawling up the tree, and it doesn't matter whether or not there are any nutlets on the tree. When the second flush comes out, if there are no nuts on the tree, they don't climb up there. And of course, his first thought, because he's a retired entomologist, is there has to be a pheromone responsible for this. But I kept saying, I don't know. There's so many other chemical cues that could be at work there. Well, I'm now wondering, thanks to this presentation, well, maybe it's a volatile coming out of the frass. Maybe it's actually a volatile being released by the decomposition or by the frass itself, that there is a trail that actually stops them from where that frass must be falling and hitting the ground, that they can tell if there's no frass there are no nuts there. So why would I climb this tree? So that's going to be where I take that. But anyway, thank you for your time. I appreciate it.
Thank you, Lacey. That was great. And then, you know, every one of those questions can be a Southern IPM Center funded project because we can ask and we can figure it out. Another potential hypothesis could be that perhaps there is more than one genetically differentiated population of these aphids. One adapted to come early when it usually floods. So selection-wise, they have been basically the behavior is built in that when you are that early in the season, you need to go up to survive, and it's kept through natural selection. And perhaps the second population, it has a later phenology, and the genes that explain the later phenology could be linked to this other behavior. So you could be talking about 2 or more biotypes of this weevil that occur at different times in the year, or even perhaps if they do not reproduce among them, if the phenology are apart enough that they cannot reproduce, you may even have genetically differentiated populations of this aphid, kind of like of this weevil, kind of like I show in the MLLF genetically distinct groups that have different behaviors. And that's why it's so important to look at the genetic composition of our pests, because genetically differentiated populations may definitely different behavior, like what you are telling us here, going up and down, or resistance or susceptibility to chemicals, susceptibility to natural enemies. We need to know. So the first thing I will do with that population geneticist myself is to sample them and look at their genomes and see where, if they are genetically distinct all over, which will indicate reproductive isolation and potential host races, or if they are only at some specific areas denoting diotypes and perhaps selection-driven differentiation. So, but that's just one of hypotheses to add to the ones that you proposed that could also be possible. So exciting and fascinating, and your dad never stops being a scientist. Of course, we observe things that were there forever and nobody noticed, but the scientists do, and then we have questions.
And apparently publish 9-page articles in trade magazines and have people going, no, this is not appropriate for that. Why don't you give this to extension? But yes, that's entirely possible, because there is an alternate host that would be right there for all of the major pests of pignut hickory, which produces a nut that's virtually identical to pecan. So that's entirely possible. I will certainly suggest all of this to him, and we may be in touch.
Yes, I will maybe convince one of my graduate students to take those projects, and then we will know the answer and tell your dad, it was not this, but it's this. And he can be a consultant in the project.
I did want to mention too, Lacey, so Raul talked about us having grants at the Southern IPM Center. Those will open back up again in either August or September. Can't remember exactly when, because we changed the date last year. But you do not have to be tied to a university. So even your father being retired, he could still put in for one of those grants. So I'll be sure to send you information about those if you'd like.
You can absolutely send it. I'm telling you, the work will be done, at least as far as everything that he can do himself in collecting things. It gets done anyway. So thank you.
No problem. And that's for anybody else on the call too that might be interested in doing research in IPM. So Dr. Medina, looks like that's the end of our questions for today, but we do still have a few people on. So I wanted to ask you the question that I warned you about at the beginning. If you were leaving them with one thing that they should remember from your talk, or that you want them to remember, what would that be?
Oh my God, the only thing I would like them to remember is that we have been trained to solve pest problems in a simple fashion, mostly due to chemical control industry that is trying to sell that as a product, because that's the easier way to use something. But we need to break from that and understand that there are hidden costs of those simple solutions. We call them externalities in economy, but not the things that nobody measures, like environmental problems, human health issues, toxicological effects in wildlife, and stuff like that. So we need to change that mentality of the commercialization of biology and put it more into complex solutions. Do not have simple solutions. Complex solutions have complex problems do not have simple solutions. Complex problems have complex solutions. And complex solutions look like the talk I just presented to you, when we need to know which genotype we have where, when, and how can we attack that. And is it that we need to target the genotype itself, the insect, the bacteria? Do we target the entire bacterial community, or we just target one? Can we gene edit something to change that honeydew from not attractive to attractive? All that is complex. So it's very difficult to sell, but it's the thing that will get us through sustainability. So the only thing I would like guys to remember is complex problems do not have simple solutions. Complex problems do have complex solutions. And we need to start training the public to understand that. So they start thinking more in terms of conditional probabilities, being like, you know, given this, I will do that, rather than I will always do that. And that will be a lifelong work for not only people, but institutions. That's the one thing.
Well, thank you so much. Again, thanks to our speaker, Dr. Raul Medina at Texas A&M. And if you have any questions, feel free to reach out to him, or even to me. You can find us on the web at the Southern IPM Center at southernipm.org. So thanks for coming, and we hope that we'll see you again soon.
Thank you, everybody.